We've all heard that saying, right?
Mm-hmm.
About memory.
Neurons that fire together, wire together.
Yeah, exactly. Super catchy. And it kind of makes sense, doesn't it? Like the more you do something, the stronger the connections in your brain get.
Like building a path. The more you walk it, the more defined it becomes.
Yeah, exactly. But some new research coming out of the University of Chicago, well, it's adding some really cool new details to that picture. It seems it's maybe not quite that simple for you.
Right. For a long time, we've had this model of how the brain learns. It's called the Hebbian model. It basically says that learning happens by strengthening the connections between neurons that fire at the same time. Like you said, creating that well-trodden path.
So what are these researchers saying then? You said it's not that simple.
Well, it's not that it's completely wrong. It's more like there's more to the story, especially when you're talking about the hippocampus, which is a brain region that we know is super important for forming new memories.
Right, the hippocampus. So for all of you listening who want to really get a grip on how your memory actually works, today we're going to be going deep on a new study published in Nature Neuroscience. And the researchers, Mark Sheffield and Antoine Madar, are really getting down to the nitty-gritty details.
Yeah, they're looking at exactly what's happening at the synapse level, those tiny connections between brain cells as memories are forming. And for you, the listener, this could give you a much more accurate, much deeper understanding of how your own experiences actually shape your memories.
Okay, so let's jump in. Our mission today is to really unpack this new research, grasp these findings, see what it all means for how we think about memory and learning, what's actually going on inside your head when you make a memory.
Yeah, absolutely.
Okay, so let's start with the basics. Just a super quick overview. We're talking about synaptic plasticity, right?
Right.
This is the idea that those connections between your brain cells, the synapses, they can actually change in strength depending on your experiences.
Right.
And that's how we believe we learn and remember things.
Yeah, that's the basic mechanism. Your brain adapts to new information by changing the strength of connections between neurons. It's amazing. So like we said, the traditional view, the Hebbian idea, is neurons that fire together, wire together. When two neurons are active at the same time, the connection between them gets stronger.
And that makes them more likely to fire together again in the future, right?
Exactly. And that's how you encode an association. But on the other hand, if they're active at different times, the connection gets weaker.
So that path kind of fades away if you're not using it. So what this new research is saying is it's more complicated, right?
Yeah, it's not quite the whole picture, especially when you're looking at the hippocampus.
So what specifically did they find? What's the challenge to this Hebbian model, for our listeners who really want to understand what's going on in their brain?
Well, the really fascinating thing here, the thing that really makes you think, is that they found that even after you learn something and it becomes familiar, the activity patterns of the neurons in your hippocampus, they keep changing. They don't just become fixed.
So even when you know something really well, like, say, your favorite coffee shop.
Exactly. Like you walk in for the first time, your brain is creating a map of the place.
Right.
But even after you've been there 100 times, the neural representation of that space doesn't become static.
Huh.
Mark Sheffield, one of the researchers, he had a great analogy. He said, even when you walk into a familiar room, the neuronal activity representing that room doesn't just settle into a stable pattern. It keeps subtly changing.
Wow. So it's like our brains are constantly updating the map, even when we already know the territory.
Yeah, it seems so.
But, if we already have the memory, if we've already learned the layout of the coffee shop, what's driving those changes? What kind of synaptic plasticity is going on there?
That's exactly the question they want to answer. And it's a tough one because it's not like you can just, open up a brain and watch those tiny changes happen while someone is, living their life.
Right. Makes sense to you.
Yeah.
It's like trying to watch a single ant move in a giant anthill.
Yeah, it's a huge technical challenge. But that's where place cells come in. And they're really key to understanding this whole thing. So place cells are these specialized neurons in the hippocampus. And they only become active when an animal is in a specific location within an environment.
Like their little GPS system.
Exactly. And each neuron has its own place field, like a specific spot on the map that it responds to. So different neurons will fire for different locations, and together they create a kind of internal cognitive map of the space.
Wow.
This discovery, by the way, won the Nobel Prize in 2014. It was a huge deal.
So that gives researchers like Sheffield and Madar a way to actually observe how the brain is representing space, which is like a core part of a lot of our memories, right?
Yeah, exactly. So what they did was they recorded the activity of these place cells in mice, and they had the mice explore both new and familiar environments. Now, you might think that, in a familiar environment, the place cell activity would be pretty consistent. Like the mouse goes to the same spot, the same neurons fire.
Seems logical for you.
Yeah.
Same place, same brain activity.
Right. But the really cool thing is that's not what they found. The place cell activity was actually slightly different each time the mouse went to the same spot, even in the familiar environment.
No way. So even though the mouse knew the place, its brain was still kind of tweaking the map.
Yeah, it suggests that there's this constant, ongoing synaptic plasticity, even for familiar places. It's like your brain is always fine-tuning its understanding of the world.
And it's happening in your brain, too, as you're listening right now.
Exactly.
Okay, that's super interesting. But how did they figure out what kind of synaptic plasticity was causing those changes? How did they go from, like, seeing these neurons firing to understanding the actual mechanisms involved?
So to really get at that, they used computational modeling. Basically, Antoine Madar, he built a computer model of hippocampal neurons. And then he tested out different rules of synaptic plasticity within the model.
Kind of like a simulation of the brain, right?
Exactly. And they wanted to see which rule could best reproduce or mimic the actual changes they were seeing in those mouse place cells. They compared the traditional Hebbian model, the spike timing dependent plasticity, or STDP.
Neurons that fire together, wire together.
Right. But they also tested another rule, a less studied one, called behavioral timescale synaptic plasticity, or BTSP. And you know what they found?
Yes.
BTSP was a much better fit. The Hebbian model could only really account for gradual shifts in the place fields, but the actual data from the mice showed all sorts of changes.
Like bigger jumps, maybe.
Exactly. Bigger jumps, sudden shifts. And the BTSP model could capture that whole range of dynamics.
So it's like STDP is like making small adjustments, while BTSP is like sometimes you just got to overhaul the whole thing.
Yeah, that's a great way to put it.
So that's happening in your brain too, by the way.
Right. It's like the brain is using two different tools to update the map. STDP is for fine-tuning, and BTSP is for those bigger, more dramatic changes.
Okay, that's amazing. So they're saying that this BTSP is really important. But what is BTSP exactly? How does it actually work?
Well, they already knew that BTSP is triggered by these big, rapid increases in calcium levels inside the neuron.
Calcium. What's calcium got to do with memory?
Calcium is like the signaling molecule inside neurons. It's involved in all sorts of processes, including synaptic plasticity. So what they found in this study is that while these big calcium jumps don't happen all the time, they occur more often when the animal is actually learning something new, when it's forming a new memory.
Interesting.
And here's another cool thing. Once a place field has formed, once that neuron is reliably firing when the animal is in a specific location, the frequency of those BTSP events, they start to go down.
Okay, so it's like once the brain has kind of got a handle on the place, it doesn't need to make those big changes as often.
Yeah, that's the idea. But even though those BTSP events become less frequent, they can still happen. And that's what explains why those place fields continue to drift and change over time, even for familiar places.
So our brain's map of a place is never truly finished. It's like a work in progress, constantly being updated and refined.
Exactly.
But then the big question is why? For you, the listener, why is the brain doing this? Why not just settle on a stable representation of a place once we've learned it?
That's the million-dollar question. And the researchers have a couple of ideas. One is that maybe these constant changes help the brain distinguish between similar memories that happened in the same place but at different times.
Like if you meet a friend at your favorite cafe on Tuesday, and then you meet a different friend there on Wednesday.
Right. The slight shifts in the place fields might be what helps you keep those two memories separate.
So it's like the brain is adding a timestamp to the spatial map.
Yeah, kind of like that.
And then Sheffield has this other idea that I find really interesting. It's almost like a poetic view of memory, right?
Yeah, he talks about it in the paper. He compares it to Proust, the French novelist.
Right.
Proust has this whole thing about how a sensory experience, like the taste of a Madeleine cookie, can suddenly unlock all these vivid memories.
And he thinks these shifting place fields might be part of that.
Yeah, he suggests that maybe these constant dynamics, these changes in how the brain represents a place, they're not just about the physical space itself. They're about encoding the whole experience. Like, think about it. Even if you're in the same room, the experience can be completely different depending on, like, the time of day, who you're with, the smells, the sounds.
So having coffee in your living room is a different experience than having dinner there, even though it's the same physical space.
Right. And Sheffield's idea is that all those subtle differences, they're being woven into the memory through these constantly changing place fields. It's like the brain is capturing the richness of the whole experience, not just a static snapshot of the location.
It's like instead of just a photograph, it's more like a video with all these layers of sensory information and emotional nuances. It's pretty mind-blowing.
Yeah, it really challenges our understanding of how memories work.
And it all comes back to this idea that our brains are incredibly dynamic. They're not just like recording devices passively storing information. They're constantly active, constantly updating and refining our understanding of the world.
Exactly. And that's happening in your brain right now, listener, as you're taking in this new information.
Right. And speaking of new information, I should mention that this research was funded by the NIH, the Whitehall Foundation, the Searle Scholars Program, and the Sloan Foundation.
And Anki Zhang and Can Dong also contributed to this work.
So for you, the listener, if you want to dig deeper, the paper is in Nature Neuroscience, and the DOI is 10.1038, SOR 1593-025-018946.
So to kind of sum up the key takeaways for you, this research really points to a pretty big shift in how we think about memory formation in the hippocampus.
It's not a one-and-done process.
Right. It's not like we just record a memory and then it's fixed. It's more like a constant, ongoing evolution of how the brain represents our experiences.
And that evolution seems to be driven more by this BTSP, this behavioral timescale synaptic plasticity, than we previously thought.
Right. The Hebbian model, the STDP, it's still part of the story, but BTSP seems to be playing a much bigger role in shaping these ongoing changes.
So for you, the listener, as you're going about your day learning new things, remembering past events, it's pretty amazing to think about all this complex activity happening in your brain. Like, even when you think you know a place really well, your brain is still working behind the scenes, tweaking and refining its representation of that space.
And that raises a really interesting question for you to consider. How does this constant re-encoding of our experiences, even in familiar settings, how does that affect how we experience time, how we view our own past? Like, have you ever gone back to a place you used to know well, and it felt both familiar and different at the same time? Maybe a little strange, even?
Yeah.
So maybe these findings, they can help us understand that. This idea that our memories are not just static records, but they're constantly evolving, constantly being reshaped by our ongoing experiences. It's something to think about, something to maybe see if you can notice in your own life.
Absolutely. Thanks for listening, and we'll see you next time for another deep dive.
See you then.